The Fantastic Microbiological World. Jefferson Goncalves Pinheiro Silva. The Fantastic Microbiological World. 1st edition. 2023. São Paulo. 1. The Fantastic Microbiological World. Catalog. S. 586. Silva, J.G.P. 1994. The Fantastic Microbiologicalworld. Jefferson Goncalves Pinheiro Silva. 1st edition, São Paulo. Independent publication, 2023. p. 58.14 by 21 cm. ISBN 9780850944148. Subjects. B11. Microbiology. 2. Analysis. 3. Science. 2. Presentation. Dear reader, this is not a textbook and may require some effort for those outside the field of microbiology to understand. However, it is not aimed at a specific audience, and if you like to explore different topics, you are in the right place. These texts were conceived during my training as a researcher and refer to analyses of unique scientific publications or of a topic that required multiple references for its construction. There is no chronological sequence or interconnection of the discussions. The themes connect with the title, The Fantastic Microbiological World. To preserve objectivity, we choose not to add explanations or previous indexes. Doubts, suggestions or claims, contact the author through one of his communication channels. At Backfriends. Backfriends Divulgação Científica at Outlook.com. 3. I adapted each chapter of this material into explanatory videos. Although it is not a formal class, the videos address the main points of the content, offer bibliography suggestions and, progressively, I will update the material with the clarification of doubts sent by the readers. Just point your phone, scan the QR code, and you'll be directed to YouTube content for free. I felt the need to adopt this approach, since the available contents do not provide the theoretical foundations of the discussions, as it happens in a traditional pedagogical book. I think that this gap can alienate readers who do not know the area, which is completely contrary to my proposal to contribute to the popularization of science. 4. Author. Jefferson Goncalves Pinheiro Silva. Chemical technician, graduated in biological sciences and masters in microbiology from the Institute of Biomedical Sciences of the University of São Paulo, ICBUSP. I am currently doing a PhD in the same area, ICBUSP, in the following subjects: metabolic networks, systems biology and bioprocesses. Experience and qualification. Researcher. Development, execution and optimization of bioprocesses. Statistical analysis of experimental data, preparation of reports and technical presentations. Execution of commonly used analytical routines in the microbiology laboratory. Basic and technical level teacher. Preparation and delivery of classes for students of technical education in nursing, aesthetics and pharmacy in the following disciplines: human morphophysiology, pathology, collective health, biochemistry, drug production, pharmacobotany, phytotherapy and microbiology. Monitor in the MIRAM ICBUSP Bioscientist Project, 2023. 5. Monitor in the Real Lab Day Project, ICBUSP, 2022. Organizing member of the 7-winter course in Microbiology and Applied Molecular Biology of the ICBUSP, 2021. Monitor member and organizer of the 14-summer course in Biochemistry and Molecular Biology of IQUSP, 2019. Student representative of the ICBUSP Microbiology Department, 2018. Higher education teaching intern in the discipline of Oral Microbiology for Dentistry at ICBUSP, 2018. Volunteer Professor of Biology, Reed Emancipa Itapai, 2016. Volunteer Research Advisor, Siddhartha College, 2015. Scientific Initiation, USP, Department of Microbiology, 2015. Intern at the Plant Anatomy Laboratory. Botanical Institute, 2014. Intern in the Sanitation and Hydraulics Laboratory, FATEC Bom Retiro, 2012. 6. Articles in Development Scientific Contribution. 1. Improvement of copolymer production through bioprocess strategies. 2. A review of the challenges in rebuilding the metabolic network of Burkholderia saccharide to consolidate it as a platform in synthetic biology. 3. A methodological approach to assess the quality of bacterial genomes. 4. Genome-wide reconstruction of the metabolic network in Burkholderia saccharide expands its biotechnological applications. 5. In silico analysis of antibiotic resistance and virulence factors in Burkholderia saccharide. 6. Scientific thought and study of biodegradable polymers. Of the family of polyhydroxyalkanoids. 7. Synthesis of polyhydroxyalkanoid, PHA, biopolymers by bacteria isolated from the Tiet River. 7. As a child, I wanted to become a scientist to create dinosaurs. I ended up finding microbiology by chance, inspired by a professor. His passion for the countryside made me choose this path. The opportunity to work in the microbiology department has made my childhood dream more and more possible. What motivates me now is building a relevant curriculum so that one day I can work as an educator. 8. Summary. Chapter 1 Comparison between the metabolism of Staphylococcus aureus and Vinus allochromatic, page 10. Chapter 2 Extremophiles and the limits of life, page 17. Chapter 3 Spherical shape, page 27. Chapter 4 Communication between bacteria and fungi, page 31. Chapter 5 Nanobacteria, page 38. Chapter 6 Circuits Biological, page 53. 9. Chapter 1 Comparison between the metabolism of Staphylococcus aureus and allochromatic venosium. Staphylococcus aureus R. Bacteria gram-Schram positive and natural components of human skin. On some occasions, these bacteria can become pathogenic and trigger a series of problems for the host. This microorganism is aerobic by nature, using the Embden-Meyerhof-Parnas (EMP) to oxidize glucose to pyruvate. These bacteria are also considered facultative anaerobes as they carry out fermentation. The EMP pathway is the first to obtain energy and precursors for cell maintenance in these individuals. The EMP pathway is not sufficient to satisfy the demand for precursors that constitute more complex molecules that make up a bacterial cell, such as the membrane, the cell wall, DNA, among other structures. Therefore, these microorganisms need 10 to carry out other pathways for the biosynthesis of these compounds. In 1959 it was proposed that these microorganisms would follow the EMP pathway followed Byc-Krebs cycle. However, these two cycles together would lead to the obtaining of 9 of the 13 essential precursors. In 1962, Hancock, Doss and Chatterjee suggested the presence of the pentose pathway in these species in addition to the EMP pathway and the pyruvate oxidation pathways, such as the pyruvate dehydrogenase complex or the pyruvate formate lyase pathways, both of which led pyruvate to acetyl-CoA in addition to carrying out the cycle of Krebs, in which there is the participation of this acetyl-CoA molecule or the activation of the oxidative and reducing branches. In anaerobiosis S. aureus it carries out the fermentation, producing lactic acid from the pyruvate and can also, depending on the conditions in which the medium is found, produce acetic acid. In anaerobiosis, the precursors are obtained from the EMP pathway. 11. The pentosis pathway, the oxidation of pyruvate to acetyl-CoA by the pyruvate dehydrogenase complex or by pyruvate formate lyase and the activation of the oxidative and reducing branches, since the cycle of Krebs is inactive, thanks to the absence of the alpha-ketoglutarate complex, due to the anaerobic conditions in which the medium is found. In this way, Wens aureus, when performing aerobic respiration, in addition to producing all 13 precursors, the cell is also capable of making a significant amount of ATP molecules compared to fermentation, in which ATP molecules are made only through the EMP pathway. In aerobic respiration, these energetic molecules are produced in the EMP pathway, in the cycle of Krebs, and because it is carrying out respiration, the electron transport chain is active, thus being able to promote the displacement of electrons from the oxidation of the glucose molecule to the final acceptor, which is oxygen, causing the protons to be pumped into the extracellular medium, and they return to the 12 cell through ADP synthase, promoting the synthesis of new ADP molecules. Avenous allochromatic or bacteria gram-Schram negative, commonly found in bodies of water. These microorganisms are autotrophs, carrying out a process called anoxygenic photosynthesis, since the reducing component is not water but hydrogen sulfide. In the absence of light, these individuals synthesize essential cellular components through chemolitho-autotrophic processes. These microorganisms are still capable of carrying out a process in which they use organic molecules as electron donors for their photosynthetic processes, a process called photoorganoheterotrophy. The photosynthetic process will lead to the reduction of carbon dioxide to molecules that can be used by bacteria. The metabolism starts from the cycle B. Calvin-Benson. This cycle will result in 7 of the 13 cell precursors. Fundamental. During the cycle Calvin-Benson, molecules of glyceraldehyde 3-phosphate, the semi-molecules 13, then follow some common steps of the glycolytic pathway, which will result in 3-phosphoglycerate and phosphoenol pyruvate, which can then be oxidized to pyruvate. The three remaining precursors to be synthesized are produced in the oxidative and reducing branches or organism itself. See Krebs cycle depending on the absence or presence of oxygen. Both metabolisms, chemolithotrophic, photolithoautotrophic, and photoorganoheterotrophic, carry out similar steps, the difference being the oxidation of acetyl-CoA, which can pass to the Krebs cycle in the presence of oxygen, or to the oxidative and reducing branches. The steps followed to obtain the 13 precursors are Calvin-Benson cycle, a small part of the glycolytic pathway, and pyruvate oxidation. The difference between both processes lies in the energy yields. Staphylococcus aureus is avinusilochromatic. They have different metabolisms in various ways. S. aureus, it has heterotrophic metabolism, oxidizing organic 14 molecules aerobically or anaerobically. A. wine, it has autotrophic metabolism, being able to be classified as chemolithotrophic, photolithoautotrophic, and photoorganoheterotrophic, being the base of the food chain in various marine ecosystems. S. aureus, under normal environmental conditions, uses oxygen in its metabolic processes, while A. wine oxygen is used in exceptional conditions. S. aureus obtains its precursor molecules, reducing power and ATP from the total oxidation of glucose under aerobic conditions, while A. wine the obtains precursors from carbon dioxide reduction, while ATP is produced via the electron transport chain, and reducing power is restored from a reverse electron transport chain. Under anaerobic conditions, S. aureus uses the oxidative and reducing branches of the Krebs cycle. A. wine, because they have anaerobic metabolism, these branches are always active, with rare exceptions, in which the Krebs cycle is also active when, for example, these bacteria perform 1,5 chemolithotrophy. In both microorganisms, the oxidative and reducing branches lead to the synthesis of 3 essential precursors. 4. Metabolism. References bibliography. 1. Weigel, L.M., Clowell, D.B., Gill, S.R., Clark, N.C., McDougall, L.K., Flanagan, S.E., and Tenover, F.C. 2003. Genetic analysis of A. high-level vancomycin-resistant Staphylococcus aureus isolate. Science, 302, 5650, 1569–1571. 2. Strasters, K.C. and Winkler, K.C., 1963. Carbohydrate metabolism of Staphylococcus aureus. Microbiology, 33, 213–229. 3. Núñez-Cardona, M.T., 2003. Isolation and pigmentary characterization of red sulfur bacteria from the Tampamachoco Lagoon, Veracruz. Hydrobiologica, 13, 3, 171–176. 4. Weisgerber, T., Zigan, R., Bruce, D., Chong, Y.J., Detter, J.C., Han, C., and Tapia, R., 2011. Complete genome sequence of Allochromidium venosum DSM 180T. To stop. Genomic Science 5, 311–330. 5. Franz, B., Lichtenberg, H., Hormes, J., Modrow, H., Dahl, C., and Prange, A., 2007. Utilization of solid, elemental sulfur. B. The purple sulfur phototrophic bacterium Allochromidium venosum. A K-edge X-ray absorption spectroscopy study of sulfur. Microbiology, 153, 4, 1268–1274. 16. Chapter 2. Extremophiles and the Limits of Life. Marino, Nancy et al. Living in Extremes. Extremophiles and the Limits of Life in a Planetary Context. Frontiers in Microbiology, version 10, page 780, 2019. A week after the presentation of this activity, a possible sign of life was discovered on the planet Venus. The article, Living in Extremes. Extremophiles and the Limits of Life in a Planetary Context. In literal translation, Living in Extremes. Extremophiles and the Limits of Life in a Planetary Context can help us understand how it is possible there must be life on Venus, despite the fact that it is the hottest planet in the solar system, at about 450 degrees Celsius and with an atmosphere covered in toxic gases that favor the generation of intense acid rain. This fact reported around the world shows how relevant it is to discuss the biological aspect of extremophiles and how they can help us understand the development of life. 17. Forms, so that we can apply this knowledge in a practical way in the daily life of our society. Initially, it is interesting to highlight the minimum conditions necessary for life to exist. That is, under what conditions life can thrive. The author highlights the main conditions, namely temperature, pH, pressure, radiation, salinity, energy and nutritional availability. At this point in the discussion, it is important to make clear what is considered life in science, since this debate goes beyond what most of society understands as something alive. For example, if we ask a layman what is life and what conditions would it need to survive and thrive, the first thing that would come to mind when answering that question would be to say something like an animal or a plant, and that the conditions should be close those supported by these organizations. However, as the author himself points out, these are a minority among beings. In the item, parameters that limit life, the author highlights the importance of the 18. Existence of water in a liquid state or some other solvent, source of energy, so that the biochemical reactions necessary for the existence of a fundamental living being can take place. Unit. What we call cell, in addition to the so-called building blocks. In this article, the author makes clear the need for these conditions to exist, and discusses the possibility that life exists on other planets and that these may not be chemically and structurally similar to life forms that exist on Earth. At the very limits of the Earth, there is a certain variety of chemical composition with respect to the structure of cells. Going back to the beginning of this analysis, without deviating from the main topic, where a recent discovery about the existence of life on Venus is reported, I would like to highlight the author's exhaustive analysis of the importance of water for living beings. However, in a brief search I could not find any material that could indicate the existence of water on Venus, although several studies indicate that it was once 19 present on the red planet, which is interesting. Since there is life on Venus, is it? What other thing, solvent could be using this being? In the items, acidity and alkalinity, salinity and water, activity, temperature, pressure, and radiation, it is discussed exactly how much life can support in terms of values from known microorganisms. Regarding the pH and its scale, that varies from acid levels to more basic levels. We can find living beings at the end of both spectrums. The author indicates studies that demonstrate the need to maintain a pH close to intracellular neutrality so that biochemical reactions can occur in order to supply the metabolic needs of the cell. This control can be carried out by the living being itself or by the environment that surrounds it. The author comments on the ability of some bacteria to excrete acidic compounds to keep their internal pH balanced. Some of these microorganisms, for example, are widely used in the food industry for the production of dairy products. Regarding the 20. Item, salinity and water activity, I highlight some interesting aspects, such as the ability of some microorganisms to survive in low concentrations of water and high concentrations of salts, such as the microbial community of the Cryos Basin. The ability of these microorganisms can demonstrate the possibility of the existence of life on other planets. However, although they can live in low concentrations of water, this is still necessary. Perhaps one of the most interesting and easiest elements to feel by anyone on our planet is temperature. Furthermore, temperature conditions can be easily controlled by anthropogenic action, which is why this is the most chosen parameter when it comes to microbiological control. The author points out that the temperature limits for life would be around -40 degrees Celsius to 150 degrees Celsius, which clashes with any hypothesis about the existence of life on other planets, given that these temperatures are easily extrapolated to more or less. However, it is important to highlight that the resistance of a 21 microorganism to a certain temperature is directly related to the pressure that is imposed on it. Therefore, pressure and temperature are two factors that cannot be considered separately. For example, for an autoclave process to be considered efficient, it is necessary that the temperature and pressure parameters are respected. Few beings adapt to high pressure. However, this is possible and may be adaptable through the chemical composition present in some cellular structures, such as the plasma membrane. It may not be possible to infer much on the subject of pressure, although some studies pointed out by the author demonstrate the ability of certain microorganisms to adapt to high and low pressures. It is known that pressure also affects the conditions that surround a living being, such as salinity, temperature and oxygen concentrations. That is, perhaps pressure indirectly affects the living being, influencing the environment in which it lives, and this it influences the way the body adapts. Finally, the 22. Radiation aspect of life is discussed. The Earth, as we know it today, can be read as a place with a low natural incidence of radiation due to the presence of the ozone layer. However, this was not always the case, and in the early days of the evolution of life on Earth, microorganisms had to deal with its exhausting presence. Therefore, the existence of a high incidence of radiation is not an impediment to the existence of life. Since the discovery of the first extremophile in 1969, knowledge about the semicroorganisms has grown more and more. However, it is not yet possible to establish a limit to life. Today we have data on what was found and cultivated. However, as the author points out, very little of the microorganisms on the planet has been cultivated and characterized. Much remains to be explored. Finally, the author reaches almost the end of the article with the initial approach to this analysis, on which I tried to touch the text without deviating from the main theme, which is the question of whether life can originate, evolve, 23, and survive in others' planets. In this case, I highlighted the possible existence of life on Venus due to the recent discoveries of phosphine found in the clouds of Venus. The author described the physiochemical boundary conditions of terrestrial environments and life on Earth. Delineating the boundaries of life on Earth is an important step in identifying conditions that could give rise to or support life on other planets. However, the big question is, in a practical way, knowing about the existence of life on other planets can be useful to us. In what way? The author points out 4 main points. 1. Understanding of a possible process of panspermia. 2. Understanding of the impact of human actions on life on Earth. 3. Planetary colonization by humans. And 4. The potential exploration of possible forms of existing life. In a very mercantilist way, items 3 and 4 are the most striking and give hope that large companies will benefit from the possibility. However, for Venus, perhaps only item 4 can be taken into account. 24. From a less mercantilist point of view, items 1 and 2 are quite interesting, and even item 4, however, depends on how these lives are explored. We must think, will they be exploited for the good of society or for the good of small groups? Although the author has intensively explored various studies, he points out the fundamental lack of research addressing the tolerance of microorganisms to multiple extremes, making it difficult for us to understand the limits of life. It is ironic that little has been said about Venus, and what was stated in the 2019 article is corroborated by the 2020 discovery, which states that the upper layers of Venus clouds are suitable for thermotolerant or acidophilic microorganisms, adaptation to environments with low temperature and acid conditions. Now, we will have to wait for the deepening of these studies, and soon, who knows, we will be able to expand the limits of life. 25. Chapter